As discussed a minute ago, there are spatial relationships between the ganglion cells in the retina and the visual space across the surface of the cortex. Visual input perceived from the retina is organized into visual field maps in the visual cortex. The visual field maps are also called retinotopic maps or visuotopic maps. The visual field is the total area of surrounding images that enter both eyes while they are focused on a central point. Additionally, each eye has a separate visual field called a monocular visual field. The monocular visual field is the part of the visual field that can only be seen with the left or right eye. According to Spector in 1990, the portions of the visual field or a visual angle that a normal eye of human is able to see for each left and right eye separately is about 100 degrees laterally, 60 degrees medially, 60 degrees upward, and 75 degrees downward, while both eyes are focused on a central fixation point. The visual fields of both eyes, the right and left visual field, overlap extensively to create a binocular visual field. Therefore, the binocular visual field consists of two monocular visual fields. To understand projections from the visual field, which is the left and right visual fields, may also be further subdivided vertically into nasal and temporal divisions and horizontally into inferior and superior division. This figure shows the divisions of the left and right hemispheres and their related projections to the brain. When light rays reflected from an object pass through both eyes, the surfaces of the left and right retinas receive an image of the object that is inverted, top to bottom and left to right after passing through the lens. Therefore, the image of an object in the left hemisphere is projected onto the right hemisphere of the retina, with the superior division of the left eye falling onto the inferior division of the right retina. The inferior division of the left eye falling onto the superior division of the right retina. The temporal division of the left eye falling onto the nasal division of the right retina, and the nasal division of the left eye falling onto the temporal division of the right retina. Meanwhile, the image of an object in the right hemifield is projected onto the left hemifield of the retina, with the superior division of the right eye falling onto the inferior divisions of the left retina. The inferior division of the right eye falling onto the superior divisions of the left retina. The temporal division of the right eye falling onto the nasal division of the left retina, and the nasal division of the right eye falling onto the temporal divisions of the left retina. As noted, each point in real space is represented onto some point on the surface of the retina. Next, visual information forming on the retina passes through optic nerves and cross in the optic chiasm. At the optic chiasm, the visual information from two nasal hemiretinas crosses, while those from two temporal hemiretinas do not cross. Hence, the left optic tract carries information from temporal division of the left retina and the nasal division of the right retina, while the right optic tract carries visual information from the nasal divisions of the left retina and temporal division of right retina. When visual information reach the LGN, lateral geniculate nuclei, the visual information is organized according to the contralateral field of view of both eyes. The left LGN receives visual information from the left hemifield, and the right LGN receives the information from right hemifield. The retinotopic maps on the left and right LGN then projected onto the visual cortex. The superior visual field maps dorsal to the calcarine sulcus, and the inferior visual field maps ventral to the calcarine sulcus. The posterior parts of the striate cortex correspond to the fovea, and the anterior parts of the striate cortex correspond to the peripheral regions of the retina. The extent of the cortical area represented is reflected in the number of neurons in the retina. Notice that the posterior part of the striate cortex is overrepresented compared to the anterior part of it. This is because more neurons are concentrated in the central portion of the retina or fovea than in the peripheral areas. Therefore, the convergence is higher on the posterior part of the striate cortex than the anterior part of the striate cortex. The lateral geniculate nuclei, or LGN, is the main central structure of the brain that relays visual information from the optic nerve to the visual cortex. In primates, the LGN is arranged into 6 layers. Figure shows the layers of the LGN, which are numbered from 1 to 6. Layer 1 and 2 are magnocellular layers that contain relatively large cells that receive inputs from parazole ganglion cells. Layer 3 to 6 are parvocellular layers that contain relatively small cells that receive inputs from Meigert's ganglion cells. In addition, in between the 6 major layers of the LGN are coniocellular layers that contain relatively tiny or dust-like cells. In the LGN, inputs from visual information relayed from retinal ganglion cells are highly organized. Each left and right LGN receive inputs from both eyes. Input from the ipsilateral eye or temporal retina are projected onto layer 2, 3, and 5, while inputs from the contralateral eye or nasal retina are projected onto layer 1, 4, and 6. Each layer of the LGN accommodates an organized topographic map. The upper visual field projects inferior laterally in the LGN, and the lower visual field projects superior medially in the LGN. Additionally, magnocellular layers contain neurons that are more sensitive to contrast and responsive to motion or movement, making them responsive to the where and how properties of visual information. In contrast, the parvocellular layers contain neurons that respond weakly to motion or movement but are sensitive to color vision, making them responsive to the what properties of visual information. Neural signals that are carried from each layer in the LGN is relayed through the optic radiation to the visual cortex. The visual cortex is located at the back of the head. In the brain, both left and right hemisphere contain a visual cortex. Generally, the visual cortex in the left hemisphere receives neural signals from the right visual hemifield, and the visual cortex in the right hemisphere receives neural signals from the left visual hemifield. The primary visual cortex, also known as V1, is the part of the visual cortex that receives the main signals from the LGN and conveys the main output to the extrastriate areas, such as V2, V3, V4, etc. The primary visual cortex is constituted by a thin sheet of gray matter composed mainly of neural cells body and unmyelinated axons. The primary visual cortex also has a large number of neurons, approximately 350 million neurons. The primary visual cortex is structured into 6 functionally distinct layers, which is layer 1 to layer 6. Most of the input signals from the LGN are relayed to the primary visual cortex in layer 4. Layer 4 is subdivided into layer 4A, 4B, 4C alpha, and 4C beta. Layer 4C alpha and 4C beta are the major sublayers that receive input signals from the LGN. The magnocellular layers send input signals to the cortical layer 4C alpha. Parvocellular layers send input signals to the cortical layers 4C, beta, and 4B. However, coniocellular layers send direct input to layer 2 and 3. Within V1, there are special connections to process the input signals received from the LGN. Lund reported that V1 consists of resident cells called spiny stellar cells, smooth stellar cells, and pyramidal cells that are involved in internal input and output processing. Spiny stellar cells are excitatory cells that receive and process input signals from the magnocellular and parvocellular layers of the LGN. Smooth stellar cells are inhibitory cells that process input signals locally within V1 cells. Pyramidal cells provide output from V1 to other cortical areas and subcortical structures. These cells can be found in layer 2, 3, 4B, 5, and 6. This figure shows the summary diagram of the simple circuits or interaction between neurons within V1. The magnocellular and parvocellular layers of the LGN send input signals to layer 4 in V1, which then relays the information to layer 2 and 3 via excitatory spiny stellar cells. Next, the pyramidal cells in layer 2 and 3 pass the information to higher cortical areas. There are also pyramidal cells in layer 5 and 6. Layer 5 project to the superior colliculi, while layer 6 projects back to the LGN. For your information, cells in V1s are organized systematically according to receptive field properties. This figure shows the architecture of the cortical module in the primary visual cortex. The cortical modules of V1 consist of a set of columns that are sensitive to a specific orientation and color perception. In 2005, Sinchich and Horton found that the connection between neurons in V1 and sub-areas of V2 shows that the projection are organized into two pathways of functional specialization, which is the ventral stream and the dorsal stream. The ventral stream is also called the Watt system because it processes information for object recognition, for example, color and the form, while the dorsal stream is also called the where and how system because it processes information for motor actions like locations and motions of the objects. The followings are examples of simple projections in ventral and dorsal stream, which is for ventral stream, the information from LGN sent to V1, then to V2, V3, V4, next to inferior temporal, and to the higher cortical areas. While for dorsal stream, the information from the visual information from LGN relates to V1, V2, V3, and next to middle temporal areas, then sent to middle superior temporal areas and to the higher cortical area for further processing. This is a figure from Perry and Fala 2014. This figure shows that each region has a specific role or function to send the neural information from one area to the other areas. For example, area empty in the dorsal stream process the input information of speed and direction of the object or image, while area V4 in the ventral stream process the input information of angles and curvature of the object. Additionally, Perry and Fala indicates the dorsal and ventral pathway mechanism in the study of feature integration and object representation. From this model, it shows that both dorsal and ventral areas are actually connected to each other to perform the task. To process a visual information at the cortex level, it actually has a different connectivity patterns other than we know about dorsal and ventral pathway. The connectivity pattern consists of feed-forward, feedback, and parallel. Feed-forward connection is where the information coming from low visual areas, for example from V1, from V2 to higher cortical area for further visual processing. Feedback connection is where the information flows from higher visual areas to lower visual areas, for example from V4 to V2. While parallel connection, also known as horizontal flows, is the exchange information across a visual region at the same level, which is from the within V4 region itself, within V2 region itself.
Here I would like to show the trends of publication related to vision in cognitive neurosciences. I use PubMed database to explore or identify relevant studies by using vision and cognitive as the keywords, and another combinations of keywords are vision and cognitive neuroscience. The search was done in mid-October, year 2020. First, we look at the first use keywords. From PubMed database, the number of publications related to vision and cognitive showed an increasing trend, where the researcher managed to publish for about 890 articles last year and close to 700 articles until mid of October, year 2020, which is this year. Then we look at the second use keywords. From PubMed database, the number of publications related to vision and cognitive neurosciences showed an increased trend as well, and seems plateau or remain stable or consistent start from 2014 onwards, where the researcher managed to publish in the range of 150 to 200 articles. The first keyword search seems very high as compared to the second keyword search. It might be due to the combinations of the keyword itself, very broad, which includes neuroscience, psychology, artificial intelligence, and other related areas that explore cognition. The second keyword showed a small number of articles because the search keywords is more specific to cognitive neuroscience field, which involve brain imaging methods, a review mechanism of mind, and biological processes, and other aspects that underlie cognition. But still, we need to screen the articles from the search keyword to find suitable articles for our research areas. So after you search from the PubMed database by using selected keywords, usually we need to go through from one articles to another articles, because sometimes the article showed under our selected keywords also not really relevant to our areas. So you need to be careful, and you have to have a look and read in details and try to understand whether they are really related to your areas or not. Therefore, the keyword use are essential in searching for a suitable methodology and finding relevant or related to our needs or our study areas.
In conclusion, now you can define and describe the visual system and human visual pathway. From here, I hope that you can apply this knowledge to cognitive neurosciences field. We will discuss the applications of cognitive neuroscience during tutorial. Thank you.